Welcome and thank you for joining us for the HRI seminar today. I want to begin by acknowledging that we're on the traditional unceded territory of the Clay Nations here on the Prince George campus. And just if everybody takes a moment and really reflects on what that means for you, living on these beautiful lands, and being able to do our research here in Prince George. So this is our HRI seminar series planning committee. You're familiar with this all now. I just, again, want to thank Leanna and Bree for doing all the organizing and putting this all together for today. And of course, our financial contributors, which are the Sun Life, as well as the Faculty of Human and Health Sciences. And I should also note that this, our guest speaker today is supported through the Ignite Inspire Fund through UMBC to try and build some collaborations across the province. So if you haven't done so already, please check out the HRI Connect portal. So this, you can go on there, have a profile, see other researchers at UMBC, and maybe you have some common interests. And then also, just remember UMBC is doing a strategic plan refresh. You may have seen this in your email inbox, but please take a moment to do this. Health research, we want to grow here at UMBC, so your voice is important. And one upcoming event this year, this is through the Knowledge Synthesis Center.
Yeah, it's next Thursday.
Next Thursday, where they're doing a workshop about scoping systematic and rapid reviews. It's hard to see.
Yeah, it's not sharing. It was, but now it's not. Or the slides stop sharing.
Okay.
The knowledge synthesis seminar is online only, but really good for anybody interested in
So watch for the announcement's
Yeah.
gonna come up with all the information. Okay.
Yeah. Yeah, back up.
So it's my pleasure to welcome Dr. Brian Christie here to give a talk today. He is from the University of Victoria. So I had, I guess, pleasure of first meeting him in person at the Canadian Neuroscience Conference this past year in Vancouver. He is the first research faculty in the Island Medical Program, and he has really built a very successful and strong research program looking at plasticity in the brain. So that's how our brain kind of changes in response to the environment around us. Today, he's going to talk specifically about his work in mild traumatic brain injury. So I am told this is the first public presentation of this data, so you guys are all in on the up-to-date happenings at University of Victoria.
Good. And it's actually everybody's students.
I'm sorry if I don't.
All right, well, she gets that up. I'll just say thank you so much for inviting me up here. I'm Brian Christie. I was born and raised on Treaty 7 territory in Alberta, just west of Calgary. And now I'm living in the Coquitlam speaking nations territories of Vancouver Island. And that's where the majority of the work that I'm going to present to you today was performed. Although I'm going to go on a little bit of a history loop just to get started. But I did want to tell you that since starting at the University of Victoria, a lot of my research has been driven by my students' interests. I've become more of a facilitator rather than a director of research. And so we've gone into, I originally started with exercise, enhancing learning and memory performance in the brain. And we'll talk a little bit about how this can change cell numbers in the brain and increase plasticity. But then some researchers at UBC got me involved in FASD research because it ends up being the diametric opposite of everything that exercise does to your brain. And one of my first translational programs was looking at how we could use exercise in children with FASD to help improve synaptic plasticity early in life. Of course, living in BC, we also got involved in cannabis research. And I have some of the purest THC that you can in our little lockbox. We won't be talking about that today, but it's one of the, I guess, highlights for a lot of people will be that we haven't observed a lot of significant negative effects with prenatal exposure so far. I do remember being a graduate student talking with other graduate students who were doing cannabis research in the 1980s, and they couldn't publish negative results. They had to find something bad about cannabis to be able to publish. So the world has changed a lot. And then finally, I'm going to talk today about understanding and developing treatments for traumatic brain injury. And I'll kind of take you through the origins of this story as well. It really began when I was doing my postdoctoral work at the Salk Institute, and I was working with a guy named Terry Sinowski. And my idea had been to become really good at computational neurobiology, and so to bring math into neuroscience. And I found it incredibly boring. And I started developing a collaboration with this woman, Henriette Van Craig, who had started working in Craig Gage's lab. And they had this finding where they put animals into these enriched environments, and the mice ended up learning faster. They were able to do everything better, and they had more new cells in their brains. And this idea of neurogenesis was very new in the mid-1990s. The idea that an adult brain could actually produce new neurons. And part of this finding was driven by the fact that they had discovered you could use bromodeoxyuridine as a marker, so it gets in thymidine residues in DNA, and any cell that's undergoing mitosis can take it up, and then you can go on antibody label against it. So all the little red dots are new cells in an adult brain, and all the green dots are more mature cells that are already in that layer. So if you think about this process, you have to not only get these cells born, but they're going to have to migrate and integrate into the existing cellular architecture. And that was where I entered the story. They're like, how do we know that these are functional units in the brain? They aren't just little mini tumor cells that are growing randomly. And so we worked with a guy named Theo Palmer, who introduced GFP, green fluorescent protein, as a viral vector that could be expressed by any cell undergoing mitotic division. So then I could go in and look under the microscope, and I wish it was this pretty. It wasn't this pretty when I looked under, but I could find glowing green cells and then do whole cell patch clamp analysis of this. I could also do field analyses, and a field is actually measuring the synaptic activity of hundreds of cells in this local area at a time, rather than just one at a time. We did both for the original work. And when you measure the electrical activity, you have a stimulus artifact. So this is when I pass electricity down this electrode, and then it activates the axons that are flowing past. And so here's where it flows past. It's called a fiber volley, the F. And then you release neurotransmitter, and it opens up the receptors on postsynaptic cells, and that gives you your EPSP, or excitatory postsynaptic potential. And it's actually the slope of the EPSP that tells you the strength of the connection. So if you have a really strong connection, it's a really sharp slope. If it's really weak, it's a really shallow slope. Okay, and it basically, in quick summary, is the louder it is, the more receptors are being activated, and the quieter, the less receptors are being activated. Now, we can also graph this. So normally, if you measure EPSPs, I'm going to guess how many people have electrophysiology experience here. Well, yeah. So I was thinking it's probably not super prevalent here. So if we stimulate a slice over and over again, you can measure the slope of this EPSP. Oops, really quick. You can measure the slope of this EPSP over and over again. It'll be pretty stable for hours if we wanted it to be. But if you apply a small burst of high frequency stimuli, to that electrode, like 100 pulses within a second, that's over with. You can actually increase the size of that EPSB, and this is called long-term potentiation of synaptic efficacy. And we think this is how neurons in the brain increase their ability to talk to one another. So this is raising your voice. You can also give low frequency stimuli to synapses. So this could be 1 hertz, for 15 minutes in this case, and you decrease the size of that response. So you can weaken. And if you want to think about this in terms, it's kind of like if you're tuning in the radio dial. And if you're off, you've got all that chattery noise. And when it's right on, you have low signal to noise ratio, right? So the signal is very clean and you don't have any distractors. So that's one way of thinking about LTB and LTD working together. You can also think about LTB as the formation of new memories and LTD as taking them away or helping them degrade in some way. Okay? So there's lots of ways that you can use synaptic efficacy, and those are only 2 of a multitude. So we're wondering if this neurogenesis actually produced functional cells. So we had discovered, already that exercise alone was sufficient to increase neurogenesis. We didn't have to have the big enriched environments. And we looked at LTP, and this was a really unusual form of LTP. It's one you don't normally see because there's no STP. So normally you have the first pulses are a little bit higher than the subsequent pulses. So this is our control animals in blue here. And the guys who exercised showed about the same amount of STP, but then their LTP kept growing and building over time. And when I first got this result, I was kind of like, this doesn't look right. It doesn't look like LTP is supposed to. And I didn't want to publish it. And I actually ended up missing out on a Nature paper because I was worried about the result. And so we repeated the entire set of experiments again, but still got a PNAS paper out of it. So it was still a good quality journal. But I felt that it was important to figure out what was going on. And the answer didn't come up until I started my own laboratory. But I'll tell you, we got the answer. The other thing that we noticed was that these animals who were engaged in voluntary exercise had a lot more spines on their neurons for all of their neurons, not just the new cells. And so the idea was there had to be something that was being released that was causing both existing neurons and new neurons to be more plastic. So what was that? It ends up being BDNF, or brain-derived neurotrophic factor. And it was in this paper, as my first paper is actually at South Dakota School of Medicine at the time. We just had our first child. We wanted to go to more of a rural site. We didn't want to be in a big city. And it was really hard coming from the Salk Institute to get people at small universities to take you seriously that you wanted to come there. My grandparents were from South Dakota, right in Vermilion, actually, which is where the School of Medicine is. And so they thought I had some sort of family allegiance and bought that I would come there. And I did. I went there. And we ended up doing this publication. Joel Farmer is now an anesthesiologist in South Dakota. So he stayed in the medical field, but he importantly got this result showing that brain-derived neurotrophic factor was elevated in these animals that exercised. And why do we care about that? Well, BDNF is it is a growth factor, but it not only promotes neurogenesis, but it increases the number of spines on neurons. It increases the mobility or the plasticity of the brain. And it's something that we think is dysregulated in neurodegenerative disorders. And so it's a target to actually increase for a lot of neurodegenerative disorders. Okay. So we have this is all just the prelude to my talk, right? So hopefully I'm getting up to where I was when I started doing my next phase, which is brain injury. And this is also where serendipity in science comes into play. This is actually a painting called Serendipity by Galina Zimatori. I had gone to a conference and I was presenting my results. And at the conference, I met this individual, Jocelyn Flaubert, who was talking about video games. And he saw my work on brain-derived neurotrophic factor and exercise and came up to me and said, do you think my game is increasing levels of brain-derived neurotrophic factor in the brain?
You know, I know that it takes a lot of attention, it uses memory, visual processing. We really use these overload principles, and it's also continuously adaptive and challenging, no matter what your level. If you're really bad at it or you're really good at it, it constantly challenges you because it uses this step gradient where it makes you only get 50% of them right. So if you're really good, it makes it even harder. So you're going to get 50% of them wrong. If you're really bad, it makes it really easy. So you're still going to get 50% wrong. So you're always going to be learning. You're always going to be pushed no matter what your skill level. And he thought this overdrive was the important part of the game. And the game at this time was being used by a very specialized population. And I asked him, I said, well, can I see it? And he goes, sure, let me set you up. And so, first I'm going to show you what the game is like.
You can go back.
Can I? Oh, I'll show you what the game is like. It's more fun for everyone. So we're going to have 4 of these balls are going to have a little glow around them. These are your targets. I'll move this so that you guys can see. Oops. And now I'm done my talk. Can you guys see?
Yeah,
okay?
we're good.
Okay. All right. So 4 of these targets are going to glow, and you're going to try and track them. The other 4 targets aren't going to glow. Okay. Now, the problem is when you're trying to track them, they're not going to have the glow around them. So all of them are going to look the same. See if you can pick.
Where are you supposed to fixate?
You can fixate on the green dot if you want. That can help you. Some people do, some people don't. There's our targets. It's only for 8 seconds. Okay, now you see all the balls light up with numbers. Okay. So now figure out which numbers you think are the targets that you're after, and the game will show you. So here's the person selecting what they thought were the numbers, and they got it right. All of them blinked up. So now the game goes faster. So these are, see, it's not crazy fast right now, but this is the game. Okay.
Is this on easy level?
It depends on who you are. So when we went to see the game, I guess I can't do it this way. We found out the only game in the province of British Columbia was owned by the Vancouver Canucks. And so at the time I'm coaching hockey, my 10-year-old son, you get the idea, you love the idea of going to the Vancouver Canucks and seeing them. They were fantastic hosts, and they invited us in, you gotta see it. And while we're seeing the game, the stadium and everything, I'm talking to the trainers, and they talked about how the athletes really loved it, except for the ones that couldn't do it. I was like, oh, who can't do it? And they're like, well, the guys that are out with injury. What kind of injuries? They were all looking at players with concussions. They couldn't do it. At this time in the world, it was really becoming a point of interest how to diagnose concussions. And there was a software program called Impact that was being used. I played a little bit of football in my day, and I know that athletes tend not to want to be pulled out of the game, and they might take a software program like Impact and do really poorly before they're playing in a game, so that if they get a concussion and they're doing poorly, there's no difference, right? So there's a way to fake it out. This is a game that offered a lot of possibilities because it makes you, if you don't try, you get a very flat learning curve. Nobody wants a flat learning curve. That means that you're not a very good athlete. So everyone wants to do well. So there's this impetus that's also involved in the game. And so we were wondering, could it be used as a way to look at concussions? And I had a really strong team. There's actually a lot of kinesiologists involved in this in the beginning. There's the team when we're going around and doing a lot of baseline testing with athletes and then looking for them to get concussed. It ended up taking us several years to get enough concussions from our baseline population. And at that time, I ended up having Melanie and Taylor in the laboratory who are really adept at statistical analysis. And they went through all of the data from all of our subjects and found out it was only the individuals who were symptomatic at the time, just following a concussion, that were impaired in being able to do NeuroTracker compared to everyone else. So if you were asymptomatic, but it had a concussion in the prior history, you didn't do significantly worse than control subjects who had no history of concussion. We also came up with a lot of other interesting little findings, like individuals who also learned to play a musical instrument did phenomenally well at this task. Individuals who are good at math did phenomenally well at this task. And athletes that played sports that involved having to look at wide areas of the field, soccer, hockey, football, lacrosse, did phenomenally well at this. So we love testing the rowers. And it was really funny one day when actually it was a swimmer ended up getting the highest score of all the athletes, because there's no visual perception in the swimming, right? It's just put your head down and go straight forward. Taylor was a swimmer, so that was pretty fun. So we had this idea that it was involved in, it could be a benefit for concussions, and we're wondering if it was also something to do with having poor learning and memory processing, sequelae of concussions that could be involved. And I was lucky enough to have two neuropsych students that had ended up in my laboratory. Julie Irwin's faculty advisor had retired mid PhD, and Stella Stiana just showed up on my doorstep. She was from Italy and had to go out and do a research component for her PhD thesis. And so they ended up helping collect a lot of data. And we looked at older subjects without concussions, but ones who had subjective memory complaints, because we were looking to see if it was memory deficits that was driving the differences in the game. And they had a pretty good sampling of aged individuals, 60 to 69, 70, 79, 80 to 90. And part of the reason we went with aged subjects is I was starting to think, I'm getting old. I had more than a few headbutts. I put my chin through a car dash, and yeah, I wonder what's going to happen with my brain as they get older. And I think a lot of people as they age start thinking about, what about those younger days? And there are also a lot of reports in the literature at this time about pro hockey and pro football players who had had significant numbers of concussion, who had mental struggles. And they believed that there was this new disorder called chronic traumatic encephalopathy. Well, so what surprised us was all of her group was able to perform the task and learn, but they also showed improved scores in cognitive testing. So these are clinical psychologists who did clinical testing before individuals started the game, and then after they did a 10-week session with the game. So they do it twice a week for 10 weeks. They had improved scores in memory tasks, and so there's a variety of neuropsych tests that they use, working memory tasks, cognitive flexibility tasks. And what was more astounding to us, and this only came because a reviewer didn't believe our results, they said, can you go and test these subjects again to see if there's any permanent change? And so by the time we got the reviews back, it had been, I think, 5 or 6 weeks. And so we recontacted all of our subjects, and they all agreed to come in and do the neuropsych assessments again. So we got them all at least 7 weeks after the training was finished. So, 2 months afterwards, it persisted. We actually thought it would die down because they weren't engaged in the training, but they had persistent increases in cognitive flexibility, working memory, and memory, long-term memory scores. So this really changed our attitude about NeuroTracker. It wasn't just going to be a tool to identify who had a concussion. Now it becomes a potential therapy. Got ahead of myself there. Just wrote that part this morning on that. So at this time, the pandemic had hit. Things slowed down. Taylor had just started her PhD at the beginning of the pandemic. She ended up doing a lot of statistical training during the pandemic. Became very adept at that. And one of her proteges was Jamie Morrison, who ended up coming on and doing a master's thesis with me at the beginning of this last project. I'm going to show you. What was even better is Taylor's project we used as the basis of a big grant. And here's their projects. History of concussion projects is Taylor's, so they're looking at concussions in older individuals and seeing how that affects their performance, and if we can use NeuroTracker as a therapy. And then Jamie worked with Victoria Brain Injury Society, and she looked at individuals who had much more severe brain injuries, so moderate to severe brain injuries. But their work was used as the basis for a grant that we wrote to the CQDM Foundation, which is sponsored by Brain Canada. And I guess just over a month ago, we got 2.1 million to do all of this work. And work is being shared between the University of Montreal, the Justin Flaubert's laboratory, and our laboratory in Victoria. And it was a strange grant. It required that you had somebody in Quebec and somebody outside of Quebec apply for the grant. And because of our existing collaboration, we ended up ranking first out of 47 for the thing. So we're pretty psyched, and they were pretty psyched about it, of course, because they also got to go to this last Canadian Concussion Forum, and everyone loved their research so much that they got free hoodies. Hoodies are a thing with us. And graft is a thing for all graduate students, right? Any free stuff's good stuff. So for the work I'm going to talk to you about now, it's all going to follow this basic project timeline. We're going to do some baseline testing. I'm going to introduce MRI analysis. I'm going to introduce blood analysis, and then we're also going to do the neuropsych assessments. Then the individuals are going to go into a 12-week intervention. They do this twice a week. 12 weeks. I won't talk about it as much, but some of these individuals also did exercise. We're looking at ways that we can increase BDNF. That's the idea, right? And then we're going to do a follow-up. Then we're going to do a 6-month follow-up. This is why it takes so long. It's a full master's for Jamie and a full PhD for Taylor. And I'm going to start with Taylor's work. Who's looking at this target population, adults greater than 50. We couldn't do the stratified age groups for finding people with a history of concussion that would come in. And actually, it ended up being harder to find people who hadn't had a concussion after word got out. We had like 12 people at one point who hadn't had a concussion, and 72 applicants that had a concussion that wanted to be part of the study. So we were begging everyone to bring in their spouse or whoever, that didn't have a concussion. And we also had a criteria that they couldn't be diagnosed with a neurodegenerative disorder or a pre-existing stroke or something like that. We really wanted to look at this history of concussion angle. And I'll explain why we have Taylor there. She made this figure, just by the FYI, with a little koala. But first thing I wanted to show you is that even with the most severe TBI patients, you still have learning occurring. And this is this learning curve I wanted to talk about. You're asking, what's good, what's bad. So an average adult is going to end up somewhere in between 0.5 and 1. If you're a graduate student in the audience, you'll probably end up more towards 1 here. As you get older, your ability to adapt and learn new things falls, but it doesn't mean this is an asymptote. This could keep rising. In young individuals, they'll have a much faster learning curve, and then it'll asymptote up here. And if you're a high performance athlete, you might end up somewhere around 2. And I used to show a video of the balls bouncing around at 2, but it just became pointless because nobody could follow it for more than half a second. Right? It's, you have to build up to that type of speed. But one of the other things that's noticeable is that there's a lot of variance in the TBI group that you don't see as much of in the healthy, no concussion individuals. And that really reflects the variance in injury type. We have people who come in with car accidents that have been blindsided by someone hitting them in the back of the head with a bat or a metal pipe, or people that have been hit by rocks, all kinds of different types of injury, right? TBIs tend to be the snowflake injury. There's so many different permutations, combinations. So having that was beneficial. And then the koala bear is because to really get a handle on the blood analysis for this, we started working with a group at Monash University in Australia, and they had pioneered a single molecule oligonucleotide assay, or SAMOA, process for looking at athletes that were in the Australian Rules Football League. And they wanted to have this quick assessment method in that league because, I don't know if, has anybody watched Australian Rules football?
Yeah, it's
Yeah.
way better than American one.
There you go, yeah.
Well, they don't have as much, actually.
No.
There's no equipment.
No padding.
Yeah,
Equipment.
yeah. And they leap, and you can tackle and hit people while they're in the air leaping, right? So there's lots of injuries. In fact, these guys often study two injury models, so where there's a break and a concussion. And it turns out the peripheral inflammation from a break or, damage to your knees or joints compounds the effects of any concussion, and you get more biomarkers. And so that's where they really become adept, and Quanterix, who designs the plates for these machines, uses them as their test bed. And so they have ultra sensitive test arrays for looking at cytokine production in these individuals. And we wanted to look at cytokines because we were wondering if part of this delayed effect, this progress to neurodegeneration that you might see after early concussions in your life, may be due to a constitutive increase in neuroinflammation in your brain throughout your life. And the answer was no. So this is good. I breathed a little sigh of relief here. I did have changes in my GFAP, IL-10, BDNF, TNF alpha. None of them were elevated or reduced compared to control. So those early accidents or early contact sport that you're engaged in wasn't producing these significant changes that you're going to see over 50. So those are it. Now, the interesting thing for us was neurofibrillary light has become a protein that has been identified as being a good predictor for someone having had a concussion in the acute phase. And what we found invariably was that people that had a history of diagnosed concussion or a history of a reported concussion, so that's coming from someone besides themselves, they witnessed them getting a concussion, but they didn't go in and get diagnosed. Very common for my generation. I don't think I ever got removed from a game, taken out from a game for having a concussion. I was removed once for half because somebody stepped on my neck and I lost feeling in my legs and arms for a moment. Then I was feeling fine. I was more pissed off that I couldn't go back in and play at the time, right? And that was the generational thing. We weren't wise enough to know that we could be damaged ourselves. So, but why they have this reduction in NFL right now, all of us, and even all the researchers at Monash are kind of like, we're not really sure. We need to start looking at how this aligns with changes in other proteins. And so this is where Taylor's statistical analysis expertise is going to come in really handy, because she's looking at multivariate analysis between not only these cytokines, but other markers like MRI analysis. So she can look at changes in individuals paired with what their actual individual MRI looks like. And so to do this, we teamed up with Dr. Jody Gowerluck, who's a neuropsychologist at the University of Victoria, who specializes in doing MRI analysis. And we came to an agreement with West Coast Imaging, which is in Victoria, to get all of our subjects in to do this baseline imaging. And then Taylor learned how to do MRI analysis to expand on her PhD project. And so this is kind of what we expect to see in everyone. Here's your neuronal control, there's no degeneration. And then, poor old Brian is going to come along and he's going to have all these degenerated plaques in his brain because he's had too many concussions. And to do the analysis, you do something called fractional anisotropy. And the easy way to explain it is isotropic motion is kind of like the wavelets on the ocean. So the water can be seen as flowing in any direction in any way, whereas anisotropic motion is always in a line. And so what we want to do is, or what we want to see is water molecules, or the vibration of hydrogen atoms with the MRI moving back and forth in sort of a linear fashion. That indicates that the axons are intact and they aren't broken. And when they don't move that way, when they're more isotropic, that means there's a break and the water molecules can move unconstrained. So where there's damage or a break. So here's one example. This isn't vibrant. And this is going from the back to the front. And what you're going to see, or sorry, that was front to back, is you're going to have some little red lines start poking up. This is where there's this reduced fractional anisotropy, right? So the water molecules aren't moving linear. And here's just from on top, the same brain, just going down through it. And you can see there's a lot. And what's interesting is it's all sort of around the center of the brain.
But it looks like he's really close.
And that was interesting to us because, sorry, one more here. When we started doing group data in compiling it all, what we found was that these injuries tended to be really focused in the middle of the brain. In fact, there is a huge site in the fornix that came up over multiple subjects. And it was also related to levels of one of their cytokines, p-tau217, which is a predictor for Alzheimer's disease. So now we think we're onto something. So we'll know more in a couple of months when Taylor's finished her analysis. But just the fact that this is in the middle of the brain, raised a little quandary for us. That's not what we've been taught. That's not how concussions are supposed to occur. And so when you get unexpected data, you need to look at it with new questions, new perspectives, new possibilities. That's an old quote from Albert Einstein. So here's the CDC version of what was happening in a concussion. And it's the old coup contra coup injury. So you have your head hits something, you get damage to the front of the brain, and then the counter force gets damage to the back of your brain. So it kind of views your brain as like a pea rattling around in a pot. And for anyone who's dissected a human brain, it's very compacted, surrounded by the meninges. They're fluid filled, really buffered. It never bounces around like this in your head. Not at all. So when we looked at the existing animal models of concussion, we can see how that philosophy is generated. When it has face validity, kind of makes sense, right? And it sounds good. Oh, you got a coup contra coup injury, don't you? Right? Sounds cool. Yeah, man, I got that. I don't have a brain injury. I have a coup contra coup. You look at animal models, and most of them have been using open head injury. And there's a lot of problems with that. One, not many people on sports fields or falling when they're walking, when they're older, etc., have open head wounds. They're usually closed head wounds. Second thing, all of these animals are under anesthesia, which, if you have a severe injury, is neuroprotective. You anesthetize someone to help reduce the inflammation, reduce activity. Another problem with anesthesia is if you anesthetize an animal and you aren't really monitoring it, their internal temperature will drop quite rapidly. And this change in temperature can trigger tau hyperphosphorylation, which again is a predictor for Alzheimer's disease. So you can actually set it up to make it look like you've got damage on the surface of the brain, have tau hyperphosphorylation just because of the model that you're using. Not to mention the fact that they're invasive, they involve craniotomy, they usually directly impact the cortex. So this is the most common model is probably the fluid percussion model, where they actually attach a little indwelling cannula to the brain. They inject a bolus of water into the brain, which compresses the cortex. The second most is the controlled cortical impactor, where you actually have a steel rod that actually directly impacts the cortex. Both of them result in damage to the brain. And so here is just a figure from Zhang et al. And reviews in Nature Neuroscience, showing the extent of dead cells after a CCI model. Okay, it's not what you expect to see in concussion. So in Canada, there's actually a lot of groups that recognize this issue. There was us, we developed this, we're going to call it the awake closed head injury, or the AKI model. But Ted Allison up at the University of Alberta also developed a pressure wave model that he uses in amphibious models. Rochelle Maciasiak and us also worked with this lateral impact model, and this is the side view or side accident car accident model. And you actually have a projectile that comes in, it hits a plate that's next to the animal's head, and the animal then goes spinning around, so you get that centrifugal force that you often have with injuries that cause concussions. And then Cheryl Wellington has a similar sort of thing. It's called the Chimera. Cheryl Wellington's at UBC. When you hear you anesthetize the animal and you suspend them upside down by their tail, and you have a rod that comes up and hits them, and then they go line up and come back down. So these three models all involve anesthesia, so they still have that problem of it's neuroprotective, and it could cause an issue that you have. This one doesn't. So we've adopted both these models so we can compare what's going on. And so the first one, the awake close head injury model, this was actually something a student that was a horseback rider, Alicia McConey, was interested in doing. She got involved in it and spent the first year of her PhD putting this through animal care. We had to make sure that all of the, safety considerations were in place. We piloted it. We showed the procedure in front of the animal care reps to make sure that we weren't causing undue pain. And in fact, you'll see the model in a second. After the animal receives a concussion, you can release them and they wander around, and you wouldn't know that anything's wrong unless you do a neurological assessment of them. So I'll talk about. So is everyone okay with seeing a concussion? This is the awake close head injury model. And what we're going to do is we're going to lower the piston down to this little 3D helmet we printed and done. So we call this the headbanger model because you're really just rapidly accelerating the head, right? When I had hair, right? That'd be a better demonstration, right? So, and you can do this 42 times and the animal will still pop out of the bag. It's not going to be unconscious.
Sorry, what kind of animal is this?
This is a male spray dolly rat.
Okay.
So that's the head. And so it's in a restraint bag because it's awake. And it's, so we habituate them to the restraint bag. So they'll crawl in. And then we do this. And sometimes I wonder if it's because they do this rapid acceleration. I don't know if you remember when you're young or when you're still young and you get to a concert and you shake your head. Sometimes you feel a little bit dizzy. And you kind of, maybe they forget that they just had this experience because they crawl up and they'll crawl up and they're quite happy until you put them on like a narrow beam or something like that, where they have to show fine motor dexterity. Then you can actually see deficits. Or we also would do this an auditory stimulus where we clap to get them to startle, and animals that have concussions don't startle. So we start grading their startle where they twitch their ears, have a jumpy motion, etc. And we have a graded list of things that we do. And so Alicia and Ryan Workman, who's now a dentist in Chimo, and Katie Neal, who just went over to Ireland to become a doctor, worked on these neurological assessment protocols for several years to perfect them all. And all of those are actually available on this current protocols in neuroscience publication, or there's a Joe video too that you can watch that shows them all. If you're interested in them. I thought I'd better move along to rather than show the results, but all these animals show signs of concussion. And so now we had to, think about what we saw in the human population and what we expect to see in the animal models. And if we have a contra-coup injury, rolled ahead too fast. If we have a contra-coup injury, then we should have damage at the top of the brain and at the bottom of the brain. In fact, one of Alicia's biggest arguments in her committee meeting as a PhD student was with a faculty member who was convinced she shouldn't be looking at the hippocampus, but should be looking at cortex because that's where the animal was being hit. Why would you look anywhere else? And if you're thinking about it a little differently, you might think that like a golf ball almost, the center of the brain could be denser, but also has more ventricles around it, so it can be more fluid and move a lot more. And so then you have this diffuse injury or center of gravity model where all the damage may be around the middle of the brain, like we just saw in the MRI. So here's again the damage that's induced by the fluid percussion model. And so Katie Neal, as she is finishing, and we had an incoming student, Hannah Reed, started looking at the brains of these animals, and it was pretty obvious that there was no significant damage in the brains. And so these are just unfiltered images of the brains before they're microdissected. And then we've done some crescent violet staining just to show that there's no damage to cell regions in these brains. So now we've got an injury that looks a lot more what you see in human concussion models. So how about the injury pattern? Well, in normal animals, if you remember that first story I talked about with exercise and neurogenesis, you're always going to see some cells that are going to be created in normal animals. And this is that process of neurogenesis. And you can identify them by giving the animals bromine deoxyuridine or BRDU. So we have the idea, let's look, see if we get more mitotic activity in the brain by giving our TBI animals bromine deoxyuridine. And sure enough, we did. I don't know if you can see it here. It's maybe a little dense, but there's a lot more in these brains. And I'll give you a close-up of it. So here's the center of the brain. You can see there's a lot around the ventricle, but then there's proliferative staining all around the hippocampus, up along midline and along the corpus callosum. So there's lots going on in these brains. And for Kenny's final project, she tried to identify what these cells were. And what she found doing multiple stains was that the BRDU cells tended to colocalize with IBA1 cells more than they did with GFAP, which is more for astrocytes and other type of cells. So it looks like a lot of the cells that are outside of the hippocampus are activated microglia. And activated microglia release cytokines. And so that makes sense. It's the source of the inflammation in the brain. And it's in the center of the brain, not on the outsides of the brain. That's the new, perspective and idea of it. Now, these microglia can also affect synaptic transmission and synaptic function because microglia normally in development prune spines. They don't do it as much in the adult brain. But now we have an injury, so they may again be activated and prune spines on dendrites. So we wanted to test that. And this is where some of you may recognize this guy, Patrick Nahirny, who's our histology expert at the medical program. We shared a student, Juan Tribino Paredes, and we decided we're going to try doing this dye-eye technique. And so this is an actual needle. And on the very tip of it, we've got a fragment of dye-eye that we've ground up. So we've taken crystals of dye-eye, we ground them up to make a very fine grain of dye-eye. And then we're going to implant this in different parts of the brain so that we can look at cells. And this really labels the dendrites of cells really well. And it can also be used as a fluorescent marker because it fluoresces when it's illuminated. And after that, you can get these brilliant pictures of spines in the brain. And you can look at spines in control animals and also spines in animals that have had repeated mild traumatic brain injuries. And because there's a lot of spines to count, Emily Bostashane, who ended up becoming a research assistant, and I'm blanking on Annika's name, I got Annika, ended up doing a lot of the counting for a while. And what they found was that there was a regional specificity. There were more spines lost in the dentate gyrus of the brain than in the CA1 region of the brain, where you basically didn't see any loss in our animal model. This again is different than those lateral or the fluid percussion injury or their CCI model. They saw lots of spine loss throughout the brain. Okay, so we saw this regional difference, and it was the dentate gyrus that had the most. So we started looking at electrophysiology in the dentate gyrus, and this is just to remind you again, we're going to look at the slope, and we're going to do field recordings for this work. And this work is being done by Dr. Eric E. Olson, who's a postdoctoral fellow in the laboratory. And if you want to see how he does stuff, they've been creating these online videos on our webpage that explain every technique that we use. It's kind of fun. And we're, maybe I'll show you at the end. So I also want to remind everyone that each EPSB represents the activity of hundreds of synapses simultaneously. So the first thing you wanted to know was, do we have an impoverished brain? Do these mild traumatic brain injuries impoverish it so that you don't have as big an EPSB? And it turns out no. The animals with mild traumatic brain injury can have just as large a response as the control animals. And presynaptic indications that you can look at pair pulse facilitation with also aren't changed. So they're releasing as much neurotransmitter as control animals, and the receptors are there still. So we haven't lost anything as far as we can see, even though we know spine numbers are down. The active synapses make up for any spine loss. But let's look at synaptic plasticity, and we get a different story. So here's that stable baseline. We induce LTP with megahertz. And what he found in adolescent animals is that here's our control animals that shows a great LTP, but in the animals tested 7 days after they got their concussion or 30 days after they got their concussion, they had impaired capacity for synaptic plasticity. He also did this in young adults, and he chose these two age groups because adolescents and young adults tend to be the most adventurous, whether it's mountain biking, skiing, or playing interactive sports. So yeah, and what you discovered was that, oops, sorry, with the adult brain, there is actually an acute phase where you had this reduced synaptic plasticity, but they recovered quicker. So it looks like the developing brain, the adolescent brain, is a little bit more susceptible to long-term damage. And that may be important when you think about kids going to school after a concussion. And BC Can, which is a BC initiative, actually talks about the need to educate teachers about bringing kids back into the school. And they have a great website that if you're involved in coaching or you have kids or friends that have concussions, you can look at. They have pages for parents, for practitioners, for educators, and for athletes, helping walk them through the system in a non-threatening way. So finally, out of all this, we did start off with BDNF. We were interested in BDNF. And now we had a bunch of models where we could test BDNF. And so our people that are involved in the competition are Justin Brand, who is going to work with the animal model. And he's got a variety of behavioral tests, but he's also taken blood throughout concussions. We have Jamie Morrison, who's going to work with the more severely damaged people. And we have Taylor, who's laughing at the other two because she knows that she's going to win. Why? Because Taylor is very, very competitive. And in fact, while she was down in Australia, while she was working, she also ran an Ironman and qualified for Worlds. And now gets to go to Spain this spring and compete in Worlds for a triathlon. Yeah. She tends to be that type of person. It's nice to have in your laboratory, believe me. And she also sent me back our first set of results because she did win the competition. And this is using that Samoa to look at BDNF levels in our patients. And in our control subjects, there is absolutely no change in BDNF levels. But in any individuals who went through NeuroTracker, we had a significant increase, except for this one. So basically, everyone showed a significant increase just by using NeuroTracker alone. She also did run a combined exercise and NeuroTracker package, and in the subjects that she had looked at so far, there was no difference between these two, and this is just estimation statistics showing the confidence intervals for the change that we see for each one of these groups, and so you have a high probability of this being an accurate result for the NeuroTracker alone. Also for exercise plus NeuroTracker, and exercise alone also gives you a bit, but not as much as anything that involves the NeuroTracker, surprisingly. So it looks like that effortful processing that Jocelyn Filbert was talking about in the beginning does have something going for it, and it may be producing, well, we can now say it's producing increased levels of brain-derived neurotrophic factor. And hopefully in the next few months, we're going to see what happens in the animal model and in our severely damaged people. And Taylor actually took down all of that work. And this is Jocelyn's also competitive, so he's laughing now too that we got the right result. And I also wanted to do a shout out to Emma and Jamie, Alana, who all got trained to be phlebotomists to help out Taylor with the work. She also got trained as a phlebotomist so that we could collect blood samples from all the volunteers that took part. I also wanted to put up this picture because Emma did end up beating Taylor, and she won a bronze at the Commonwealth Games as a Paralympic guide in triathlons. So yeah, Taylor tends to collect these amazing people around her. Alana just randomly decided to run a half marathon because she was hanging out with these two. She never trained or practiced and completed it, which is more than I can do. So that's the type of group that they have going for them. So it's no wonder that they won the competition, right? But I have a lot of students in the laboratory, and these are sort of pictures from over the years. And so Christina Pinar and I think Juan's hiding back here. Both helped get the NeuroTracker thing going. And then as we moved along, different people started moving into the laboratory. We also had a lot of help from Sandy Schultz, who was at Monash University, but now, because his partner is living in Victoria, has just started at Vancouver Island University. He's probably one of the most cited neuroscientists in brain injury in Canada now. He's about 4th most cited in the world. He's working at Vancouver Island University. If you ever want to meet someone or contact, he is a font of knowledge. Jody Gawryluk, MRI specialist, clinical psychologist, also a fantastic person to work with, and has just become the director of the Institute for Aging and Lifelong Health. Another great person. Pat, a lot of you may see him on video. He's the histology expert at the Island Medical Program. Also an excellent EM microscopist. He produces some of the most beautiful images I've ever seen. And Pedro Grande is another random person. He also came down from Nanaimo because his partner was living there. He's an EM microscopist from Spain who actually does some really specific immunogold labeling of receptor terminals. So we've started giving him a lot of our samples as well to do his work because he doesn't have to teach all the time like Patrick does. So anyways, and I'll leave you with just some comments from especially some of the more severely damaged individuals. Most of them have really positive things to say about the program, but there are always, some people it's not going to work for. This isn't a panacea. It doesn't work for everyone. It's not going to fix everything, but it has a lot of hope for a lot of different classes of people, aged individuals, people with light brain injury, more severe brain injury, and now I think it's going to be incorporated more widely because it's available as a distributed program that can be used in even rural sites. So we've actually got people outside of Prince George sitting in their cabins during NeuroTracker, and we send them up special goggles that turn their computer screens into a 3D screen. And this three-dimensional component of NeuroTracker is what really makes it hard because you actually feel like you're watching the balls bouncing in the box. Anyways, with that, I'll stop and give you the opportunity to wake up. And if you have any questions, feel free to ask them.
Can you handle yourself up there, or do you want me to come?
Oh, I'm happy to handle it. Yeah.
And Bree is monitoring online if there's any questions through the chat as well. So thank you so much. That was fantastic. And I will let other people ask the questions at the same time.
I saw the hand wave already.
Hi, it's amazing, it's awesome, it has a lot of potential. And I have two different kinds of questions. So at the beginning with, I guess, Dr. Mustafa and your tracker with the hockey team. You guys look at also, I mean, parents, there are, but females versus males. And if there is a difference. I know that there was something, there were a couple of things after that you looked at. And we know, I mean, basically, science, what the difference is. Is there a difference? I know there is a bit of a difference in concussion clinical science.
Right.
But how about in kind of a recovery or, is there a difference between the two, or did you look at that?
So we do, yes, and they would kill me if we weren't looking at both. And to be truthful, we were looking at women and men. But yeah, so women tend to have more reported concussions because they're more likely to go to a doctor. Men still have that hump to get over, is sort of the prevailing opinion. They don't want to see doctors. They don't want to be taken to sport. So that's a little bit of a confound when you look at what's going on. Now, in terms of our data set, we haven't seen significant differences in the reported changes in cytokines yet. But Taylor is running the multivariate analysis, so we may see something pulled out of that. In our in the animal work that we've done, we do see differences in male and female concussion changes in synaptic plasticity. In the female brain, it's much more immediate and lasts for the full period of time, whereas the males tend to have an immediate phase and then recover much faster. And we are wondering if that is due to the estrogen having a negative impact on a brain that is in metabolic crisis. And so that's kind of our thinking right now. That said, this student I was talking about who was the in the Commonwealth Games, is now it started looking at the female problem, but we also recognize that women are more likely to suffer from energy deficiency disorders. So REDS is one of the big ones in women's sport, or in all sports, but women who are in sports that involve some form of body display. So if they're wearing tight outfits, etc., tend to be more prone. And actually, Emma was suffered from REDS and was caught early enough, so it didn't change into an eating disorder, etc. So we started working with a physician in the US, Kate Ackerman, who is an MD, sports MD, who is primarily focused on women's health, but also the effects in women that are going through menopause. And so, which really fits in well with our involvement in these age populations. We have all the blood samples from them. So she's actually gonna be our consultant to tell us what we should be looking at for these. So some of that will be coming, but yeah, we're definitely thinking along those lines. And I don't know if you saw like the graph that we had of the percentage of population, like there's always a much larger female, involvement in our studies than in males. Maybe it's due to survival.
And I guess that, concussion from clinical perspective, like the, older population, obviously, and maybe more females for a variety of reasons, they are more old or something like that. Do you know, and this is not necessarily from your research, but I did, my research in neurophys, and when I had my son, I was obsessed of head injuries. Because they are just walking accidents, like, and the head first, and the body after. And I was like, and there isn't that much concussion research into, small age, right? Like from 0 to 4, they must hit their heads a lot. Mind you, parents don't necessarily go to the hospital for that, right? So there is that. And their plasticity, the brain plasticity, that one must be formidable anyway.
It was
research. Do you know?
Yeah, so and there was a call for pediatric concussion research several years ago in Canada. I think they're all Catalyst grants. I haven't seen any results from the grants yet, but that adolescent under 5 brain is extremely plastic, and it's if you're going to have to do any sort of brain surgery, you want to be under 5. You can do hemispherectomies in children under 5, and they can recover with only moderate, major, or fine motor skill deficits in their contralateral hemisphere. And actually, there's a girl that used to play volleyball against my daughter who had a hemispherectomy when she was 4, and she was a bump setter, but she was playing competitive volleyball in middle school and high school, despite only having half a hemisphere, and you wouldn't know from interacting with her. So that plasticity of the brain, extremely high young. And so that's maybe the godsend, because I also remember my daughter going off the swing head first and then bending over, and and your spine should not bend that way, right? I'm thinking, the worst, and she gets up, gotta grab. Right? Now she's an ER nurse down in the hospital down in town, right? So obviously survived, but those are. I think the women's health question is going to be a bigger focus, because we really don't understand the implications of the changes in estrogen across the lifespan for women. And as more women are getting older, understanding whether estrogen replacement is a good thing, a bad thing, or whatever, with the type of estrogen that's used, I think it's going to be, highly important, particularly when you start looking at individuals that have some sort of neurocognitive decline, whether it's from head injury or other issues. And maybe it's because of that change in estrogen levels too, right? There's a lot of, my wife's going through menopause, and she's a voracious reader, and so I get a lot of information.
You mentioned that, when you did the neurotracker with the older adults, after 7 weeks, you saw lasting effects. How long do you think those effects would last?
So we're doing this 6 month out, one this time, and we have the data, but we haven't analyzed it yet. That's, I guess, one of the losses of being able to go down to Australia and train for an Ironman is you don't have to do your analysis all the time. No, I think Taylor's working really hard on that just to answer that question. One of the other great things about this data set is we've had, tremendous retention of subjects. And I think we've only lost 17% of our subjects so far, which is really where maybe it's because we're in that pocket in Victoria where, newlywed, nearly dead, right? I'm, on the latter half of that, right? But there's a lot of elderly individuals who are, have a lot of time and are eager to get involved in research. So it's kind of, beneficial for us.
Hi, the talk was great. This is necessarily not related to your research, but have you ever thought about looking at myelination, especially because it's in the middle area and a lot of white matter regions are affected in the corpus callosum, you're seeing significant change there. And if a neurotracker would have an effect in myelin plasticity, that's something that, because I look into myelination, I was thinking, have you ever thought about it?
Yeah, I mean, I thought about it. Like I said at the beginning, I tend to get led by where the students' interests go. That said, we've got lots of scans, we've got lots of tissue. If you're interested in that. I mean, we love, enabling others to do the things, follow their passions, right? But yeah, we have thought about that, and we do see, those corpus callosum injuries, right? That fornix that's right in the middle too. Yeah.
Thank you.
Well, sorry, but, there's lots of like orthokinetic training at websites for people who have visual problems. Can those same type of websites be used for benefiting people with concussions?
Yeah, great question. And we got a push by this guy, Sam Liu, who's also at UVic in kinesiology. And so he's, using those types of programs and is wondering that same question. One of the things I do know is if you do, tests that look at like saccadic eye movement in people after concussions, their ability to do control saccadic eye movement does follow. And so you can look at King Devick tests and stuff like that. And they just have problems following, the points, or moving. And if you change the spacing in between letters, it really throws them off. It really slows down their processing speed. So there may be something there. Yeah. I don't think NeuroTracker is panacea. I think it's a task that's kind of opened up our awareness that if you have a lot of cognitive load in whatever the task is, you could maybe produce BDNF and have these beneficial effects. And with NeuroTracker, use the optic pathways, right? So you use both the dorsal and the ventral streams, but you have to use frontal cortex to kind of anticipate where those balls are going to be going to if you have to have increased focus there. And so then you've got your parental lobe, sorry, that's an old one from when I used to teach neuroanatomy, and I asked the students to identify the lobes, and one of the ones called the parietal lobe, the parental lobe at the beginning. I was glad I had a parental lobe because I had kids at the time. But anyway, so you have these, function and body self-awareness pieces. All of that plays in, and with NeuroTracker, normally we have our clients sitting. If you stand them up and change that proprioceptive scale, throws it all off, and they have to relearn again. And so it's a little difficulty. If you are a high performance athlete and let's say you want to do it skating while stick handling or dribbling a ball, or rugby players will pass the ball to each other while they're doing it. It's so that they're developing that visual awareness while they're using motor skills. And that drops down your performance level. You end up needing to start sitting and then add in other motor components because there's so much load on the system already. But that increase in load also seems to increase performance. Yeah, so hopefully that answers.
So I'm just going to step in and stop the formal part of the question and answers, but feel feel free to stick around. I'll make Brian stay here for a little bit longer. You can come up and ask your questions individually if you like. And thank you again so much for the fantastic.
No problem. Thanks for the.